Hello, I'm Akhil. This is a video demonstration of lab10, that is a lab based on GDC data. First, to perform this lab, we have to launch an EC2 instance and SSH into the instance. For that, we have to go into the Amazon Web Services portal. Here you can see that we have already created an instance which is named all diseases, which is using instance type T2.2xl, which is up and running. Now we have to connect to this instance by using SSH. I'll connect, and we have to copy this command into a terminal. Uh, I'll go into the directory where I have the temp file for this instance. Here you can see that I'm inside the EC2 instance. Now, as a part of this lab, first we have to download the mRNA data, which can be downloaded from the following website, which I have already downloaded because it takes a long time to download. And basically here we are downloading the manifest data. Uh, you can see here, here we are going inside the lab10. Here you can see that we have downloaded the live miRNA files. And now, as a next part of the data, as a next part of this lab, we have to download a tool to transfer the data, which can be downloaded from the above command, that is wget, and we have to unzip the package. That is, we after doing this step, we get a package or transfer tool known as GDC client. Now, as a next part of this experiment, we have to download the files with the GDC client. Since we have already downloaded the data, here we can see the manifest GDC manifest.txt file, which we have created using the following command. That is, first we have to give the path to the GDC client, and then specify the name of the file. And now I'll show you the programs which we have used. This is a check.py program in which we have changed the directory of the path of the GDC manifest.txt file, and in the pass file also we have changed the directory path. And here you can see that we have given a file case ID dna.csv. This is a part of the experiment in which we have to check, we have to get the case IDs related to the file. For that we have to run, for that to implement, we have to run a parse file case id.py, which we have already downloaded the case ID for the files. So here I'll show you the checksum or the integrity of the files we have downloaded by using the command. This will take a few minutes. Now you can see that the integrity has been checked perfectly and the check has finished. Now we have, as an next part of the experiment, we have to get the case IDs of the files related to the files we have downloaded, which can be downloaded using the code parsefile.caseid, which I have showed you here. And here we give the directory, that is home/ec2 user lab10, and the input file we downloaded from the website, that is a JSON file which looks something like this. And the output file is this thing named file case id dna.csv, which you can see that it will be shown here, .csv file case id dna.csv file. And as the next part of the experiment, we have to get the metadata for the files and the corresponding cases.
Hello, in this video I'll be continuing the demo of Laboratory 10. Um, in the previous part, my teammate would have shown the demo of the how to get the data files, and let's continue with the request meta file. Request meta basically is to get the metadata from the files, and here we would have got the output as the file case ID from the previous step, and the output here input would be that file, and the output here would be files meta and cases meta. In order to for this, since this is a multi-class classification, we would require a unique label. So in order to do that, we add two fields, that is primary site and disease type, along with other fields that we are going to fetch. So once the program is run, uh, I've already executed it, and we can see that we get files meta.tsv and cases meta.tsv. So using that, the next step is to run the gen mirna matrix.py, which basically gives us the CSV file which can be used for our predict.py program. So in this code, we see that we generate the unique label. Previously it was just differentiating between normal and a tumor sample. In this we have to do a multi-class tumor classification. So in order to do that, we identify the label by concatenating the primary site and the disease type. And since in since we give this to an ML algorithm, we need to get a unique integer identifier, since most of the classifiers use integer types. So we generate an unique integer for the given string using this part, and other samples which are normal are given a label of 0. So the output of this, once the program is run, is basically the CSV mirna matrix.csv. I've already run this and I've got the CSV file. The last column of this is basically the label that we generated, and the file IDs are jumbled, and hence we can see that the labels have a unique identifier here, and the max number for this is 36 upon observation, which means that there are 36 different unique labels present. Once this is done, this is the main matrix, that is input file that is used for our predict.py. In this program, we basically ran the entire code for all the classifiers and saw that the maximum efficiency and accuracy was in the random forest. Hence we went and gave the only model in the models and tuned parameters. Then we execute the ML algorithm using the fit method, and we calculate the precision accuracy F1 recall score as shown. So running this will take quite some time, and I've already done this, and we can see that we have saved a plot of it here. So this is the plot of the parameters that we find out, and the plot, instead of showing the plot, we have saved it as a figure, that is result plot. And if we open the image, we can see that the graph is plotted for the random forest classifier, and the precision accuracy, F1 sensitivity and specificity are as shown in this figure. So these are the steps that were followed in order to for the second step in the lab, to generate the matrix and to execute and carry out an ML algorithm on the data. And this is the end of the lab. Thank you.